Okay, so here we've got a section of a protein or a peptide, and you can see I've drawn 4 amino acids there in different colors. And so you can see that there's the peptide link in each case here and there's another one there. So a peptide link, of course, is a special kind of amide link, which is only found between naturally occurring amino acids. Right, now, you probably notice if you look at this, you've got lots of H's sticking out, and these H's are attached to electronegative nitrogen, so they're going to be delta positive, and of course they can form hydrogen bonds. And if you look on these oxygens, of course, you're going to have you have lone pairs, 2 lone pairs on each of those oxygens. You've also got lone pairs on the nitrogens. But what happens in proteins a lot is that the hydrogen attached to the nitrogen will form a hydrogen bond to, I'll draw a dotted line, draw a hydrogen bond to the carbonyl-carbon 4 amino acids behind. So see the purple one there, so you've got blue 1, 2, 3, 4, the black one. Okay, so you may think, oh, how on earth does that stretch that far? Well, it's because this doesn't stay as a straight line. It curls up into this thing which is called a helix, the sort of spiral, okay? And what's holding this helix together, it's called an alpha helix, or hydrogen bonds. And as I say, it's the hydrogen on one nitrogen with a 1 with the oxygen 4 behind it. So here you'd have, like, just draw it, there's NH from that residue, from that amino acid forming to, and there you got the carbonyl from the black one 4 behind it. Okay, so you get loads of these hydrogen bonds across, holding the whole thing together. Right, that's called an alpha helix. And not all areas, not all of the protein chain is forming alpha helix, but a lot of it is, quite often. So let's have a look at down here. I've got a drawing, this is on Wikipedia, there's the acknowledgement for it, that's hemoglobin. Now you can see there's lots of these spiral sections here, like that, and then what tends to happen, you get a hinge, and then you get another bit of spiral section, another sort of hinge, and then another spiral, and so on. You can see those. A lot of the protein is alpha helix. There is actually, you're wondering there, there's actually 4 chains. There's 4 peptide chains there. There's 2, the red ones, they're the alpha chains, and there's 2 blue there, they're called the beta chains. Don't confuse that alpha with the alpha helix. It's just to distinguish the 2 chains, 2 different sorts of chains in the hemoglobin. And also you can probably see that green bit there, which is the heme group, which is a non-protein bit, and that contains the iron atom, which binds the oxygen. Anyway, the main thing to get from that is that you've got lots of areas of alpha chain in that protein, and then you get these sort of, like, there's a hinge bit there, where the alpha helix breaks for a bit, and then it starts moving, forming in a different direction. So, as I say, an awful lot of proteins contain alpha helix regions. Not all proteins do, though. For example, collagen doesn't form. That forms its own kind of helix. We don't really need to know about that. But another very common thing you get in proteins is something called the beta sheet, or the beta pleated sheet. And what happens there is, rather than the peptide forming a helix, it sort of doubles back on itself. That may happen several times. And what's holding these bits together is you get hydrogen bonds across across that. And the hydrogen bonds, once again, are between the hydrogen on a nitrogen, one amino acid, and the carbonyl, the lone pair on the oxygen there, on a different one. And so you get hydrogen bonds forming across this. So that's fairly common in proteins as well, the beta pleated sheet. Right, now, as well as these alpha helixes, there's other layers of structure in proteins also. Right, so if we have a look at our AQA, list of amino acids there, this will be quite useful. Okay. Right, let's have a look at aspartic acid, and let's have a look at lysine. Now, at neutral pH in the cell, that NH2 group on the lysine will be protonated. It'll be NH3 plus, and the carbonyl, the carboxylic acid group on the aspartic acid, this one here, and that won't be involved in forming the peptide link. That's an R group. That will be ionized. That will be the CO minus group. And what's going to happen is, at various points of the protein, there's an attraction between this positive charge here on one part of the protein chain and this negative charge on another part of the chain. And that effectively is like an ionic bond, I suppose. And that holds the protein in a certain three-dimensional shape. So just to show that, you've got one. This is my peptide chain. These are all the R groups sticking out here. And here we've got, let's pretend we've got an aspartic acid R group. So that's going to be COO minus. And then on this bit of the chain here, let's say we've got a lysine, which has got that big long CH2 group. It's 4 CH2s. So I can't fit them all in, so I'll just do that. And that's going to be positive because the nitrogen is going to be protonated. That's going to be NH4 plus. And so you're going to get an attraction between those two. And that's going to hold this this peptide in that configuration there. Now you're probably aware that if you change the pH, you do it at GCSE biology, if you change the pH, what usually happens is you denature the protein. And we can explain that in terms of these interactions here. Well, imagine if you suddenly make it a lot more acidic, what will happen is that the charged group, that will accept a proton of aspartic acid, and it's going to become like that, which isn't charged. So what will happen to this interaction then? You will lose that interaction, and the peptide chain will be free to move. Likewise, let's say we make it too alkaline. Well, if it's too alkaline, then what will happen is to the lysine, which is charged, if it's too alkaline, you'll deprotonate that and you'll turn it to RNH2. This will no longer be positively charged, so you're going to lose the interaction there. So too acidic or too alkaline, you quite often end up denaturing proteins, and that's the reason why. Right, now there's another kind of interaction here between protein. They can actually form covalent linkages, and that's when you have this amino acid with the sulfur in it, which is, see, cysteine there, and we briefly mentioned it in the previous video. What can happen is, right, you've got, say this is your peptide chain, and you have these are all the R groups sticking off the side. Yeah, you have a cysteine group there, right? That's SH, and if you have another cysteine group here, another SH, there's an enzyme which will oxidize this. It will remove those two hydrogens, and what happens is that you form a covalent bond between those two sulfurs, and that holds the protein in that configuration. Okay, so just to summarize that then, what we have, I'll just show you in oxytocin, yeah, which is this little small protein here. It's only 8 amino acids long. There you have got a disulphide bridge there, holding one part of the protein to another part. Right, just summarize that then. We talk about the primary, the secondary, and the tertiary structure of proteins. Now, what we mean by the primary structure, that is just the sequence of amino acids. When we talk about the secondary structure, that's either the alpha helix regions, alpha helix, or beta sheet. And that, of course, is held together by H bonds. Right, H bonds are nowhere near as strong as covalent bonds, about 1/10 the strength, so they're quite easy to break, and that's why most proteins become denatured. If the temperature rises above 37 degrees, the hydrogen bonds are not strong enough to hold it together, and so you lose the alpha helix regions, and so the protein will become denatured. Of course, the shape of a protein is essential to how it works, and so it will cease to be a functional protein, whether it's an enzyme, whatever, it will stop working. The tertiary structure, right, now that is how regions of alpha helix or beta sheet are then arranged on a higher level, and that's held together by the ionic interactions, which we talked about between aspartic acid and lysine, and also by the disulphide bridges. And very often then you'll talk about also about the quaternary structure of a protein, and that's how multiple polypeptides are held together. And very often proteins have covalently bound non-protein groups. Such as the heme group in hemoglobin. Sorry, heme. Okay, and I think in the AQA specification, it only really talks up to tertiary structure. It doesn't mention quaternary structure, but you've probably heard of quaternary structure, particularly if you do biology. So let's just have a quick look at hemoglobin again, just to see how that. So here's our hemoglobin molecule there, right? So we've got the primary structure, which is just the amino acid sequence. Then we've got these curly bits like that there, which is alpha helix. And then we have these hinges, and different bits of alpha helix will be held to other bits of alpha helix by the ionic interactions and by disulphide bridges.